Okay, we're going to go ahead and get started. Welcome, students, faculty, guests. I'd like to give a particularly warm welcome to our SPH Dean's Council members, the SPH Distinguished Alumni Awards Selection Committee, the co-endorsees who nominated tonight's awardee, and the UW Alumni Association for their partnership in putting on this event. And I have to say, it's not on my script here, but thank you so much to Michael's family and friends. I just met someone who's like his best friend from when he was 19 who came all the way here. It's like, what an amazing turnout, and colleagues from throughout his career. It really is a testament to how amazing you are. So anyway, just wanted to say that. You weren't on my list, but special thanks to you. So the Distinguished Alumni Award is the highest award that's given by the School of Public Health. It recognizes an alumni with a demonstrated record of distinguished service and achievement in a public health discipline or setting. Tonight, we honor Dr. Michael Gale Jr. with the 2019 Distinguished Alumni Award. You'll hear from Michael on controlling viral infection from hepatitis C to Zika. And we're going to ask you to hold your questions until we have our moderated Q&A, which I'll help to facilitate after the lecture. And staff will then circulate with microphones, so just raise your hand if you want to ask a question. I'm not going to go into a huge amount of detail about what Dr. Gale will present on tonight, because I want to give him the opportunity to tell you his story. But I do want to give you a bit of background about his education and experience. So he earned his Bachelor of Science in Zoology from the University of Washington, yay, in 1985, followed by his PhD in Pathobiology in 1994, also from the University of Washington, and more importantly to us, from the School of Public Health. In 1999, after his postgraduate training, he accepted a very prestigious position as an endowed scholar in medical research and served on the faculty of the University of Texas Southwestern Medical Center in Dallas. And in 2007, we were fortunate enough to recruit him back here to the University of Washington to join the Department of Immunology as a professor with tenure, and later became an adjunct professor with both the Department of Microbiology and the Department of Global Health. During this time, he's established his affiliation with the Fred Hutch Cancer Research Center, where he's a member of both the vaccine and infectious disease divisions, and the Fred Hutch University of Washington Cancer Center Consortium. Dr. Gale is the founder and director of the University of Washington Center for Innate Immunity and Immune Disease, and co-director of the Center for Emerging Infectious Diseases. Dr. Gale has a strong commitment to training future scientific leaders. He's trained 16 doctoral students who successfully completed their degrees with him, and is currently mentoring 8 pre-doctoral students, with 2 defending their dissertation next month, which they're probably super excited about, as are probably you. And additionally, he's trained 20 postdoc fellows, all of whom now have positions in academia or biotechnology or are clinicians. And he's currently mentoring 7 additional postdoctoral trainees. Beyond higher education, the Center for Innate Immunity and Immune Diseases does outreach to local high schools and middle schools where postdocs spend time teaching microbiology and infectious disease immunology to young people. Students also visit the lab for a day to do small experiments alongside scientists, and the center also hires paid summer interns, further extending the reach of the University of Washington to our surrounding communities. If that wasn't enough, he's also editor-in-chief of the Journal of Interferon and Cytokine Research. He is also scientific founder of Kineta Incorporated, a Seattle biopharma company that develops innate immune therapeutics, and continues to serve as a consultant to the U.S. Congress upon request to impart commentary on infectious disease response planning for U.S. pandemic preparedness. He was inducted as a fellow into the American Academy of Microbiology in 2014, and since 2015, he's been on the Thomson Reuters list of the top 1% highest cited researchers in microbiology. I could go on, but we would be here all night. So without further ado, I want to congratulate Michael and thank you for your contributions to the health of our community here, but also to the world around us. Thank you.
Okay, I'm switched on. That was really nice, and I'm probably red because I'm blushing from all that.
Turn off the podium mic. Sorry, I was supposed to do this before I left.
That's good, yeah. Okay, so this is my CSI Miami pose. The University of Washington does a great job in PR, and the people from my lab who are here are probably laughing because I spend most of my time in my office, or I'm not here because I'm traveling. So this picture is a pretty good photo of what I wish I was like. So I want to talk to you tonight about the work that the group has done, first in hepatitis C, where I really started my lab in that area, and then more recently in a contemporary problem, Zika virus. These are RNA viruses that are major problems in public health. So I put this slide into the presentation just to make the point that really in the last 20 years there's been a huge emergence of virus infection. Infectious disease, of course, has a global impact. Viruses are a big part of that, notwithstanding HIV and other viruses you're very familiar with, but you're probably less familiar with hepatitis C virus because it just never got the publicity that HIV did. Zika virus is quite public, but you're probably less familiar with a lot of the viruses that are on this geographic distribution list. So what we do at the University of Washington is we study many of these viruses. The University of Washington is uniquely poised to study emerging infectious viruses, emerging infectious diseases in particular, but viruses that are very hard to handle because you have to have special facilities. And we have those facilities here, which is a very unique aspect of the University of Washington. I'm going to talk about work that the whole group has participated in, past and present. So this is my lab today, and it's a group of close to 40 people, and it's approximately 80% women. And today's National Women's Day, so I want to applaud the women and the men in my lab who've done this work. And we're a collection of virologists, infectious disease specialists, biochemists, and cell biologists who study virus infection. So to study the complexities of virus infection, you have to be an expert in everything that has to do with human physiology. I can't do it myself, but the group has expertise across the board in everything that we need to know about viruses. So as a team, we approach viruses by studying what we call antiviral immunity. So what I'm going to tell you about tonight is the work we do in this box of antiviral immunity, which is very important because if the immune response can control the infection, you're not going to get sick and there will be no disease. And we work even in a smaller box called innate immunity. You can think of this as the very first responders to infection. So I'm going to describe all this as we move ahead. But this picture shows the building we're in South Lake Union. University of Washington has a beautiful campus here you're sitting in. And then there's whole nother campus in South Lake Union. It's very specialized down in South Lake Union because the building here, the entire top floor is approximately 12,000 square feet of biosafety level 3 select agent space. It's highly specialized, probably the best place in the country right now to do biosafety level 3 work, which is a high level of containment, just a half step under, for example, Ebola virus work. So we have the capabilities and the facilities to do that work at the University of Washington. And the other picture is me holding a box of Zika virus. The reporter that came to visit us said, "We hear you have Zika virus here. We want to write a story on it." I said, "Okay." They go, "Prove that you have Zika virus first." And I said, I can't take you into the lab. And so he said, "Well, I need to see it or I'm not going to write the story." So I went into the biosafety lab and held it while they opened the door and he could shoot the picture, hoping the virus wouldn't thaw out. So that's really a box of Zika virus. And the lab's laughing too because I never go in the lab and hold Zika virus anymore. But so we study a lot of viruses. I'm going to talk about 3 of them today. Quite a bit about hepatitis C, that's where we got our start. A little bit about West Nile virus, which is a cousin of hepatitis C and came into the USA around 1999. We learned a lot of lessons from West Nile virus, and it's a good thing we did because Zika virus, which is a very close cousin to West Nile virus, has shown up in the Western Hemisphere, and it's on our southern borders, causing a lot of trouble. So those lessons now are being used for studies of Zika. We work on hepatitis B, HIV, flu, hantavirus, which has killed a couple people here in the Puget Sound area recently. Enterovirus 71, an emerging gastrointestinal infection. A little bit of work on poliovirus still, hepatitis A, and another GI virus, encephalomyocarditis virus. So I'll talk about the few, but the lessons are very similar across the board for viruses. I'm going to tell you what is a virus. It's important to know. How does the body know it's infected? So this is a fundamental question that we've been studying for quite a while. How does the body know it's infected with anything? It has to know it's infected in order to mount a response, an immune response. The basic premise of immunity is the discrimination of self against non-self. So we have a group of people that are trying to understand how the body recognizes that it's infected with a virus. I'll tell you a little bit about that. And then we'll go into what happens after the virus is recognized. Viruses make us sick because they evade the immune response, at least part of it or all of it. And I'll talk a little bit about immune evasion in terms of hepatitis C, West Nile and Zika virus. In particular, trying to link up Zika virus with how we understand the virus might cause microcephaly. I'll talk about that later. And then what have we learned? Well, I can put it all together. Maybe something useful. Can we make a drug or something that can help people against virus infection? I'll talk about that. So viruses infect us through multiple routes. There's ocular infection, there's sexual transmission, bloodborne infection, aerosol infection. All of those bring the virus into the body. And then once that happens, the body has to recognize it's infected. So here's again that fundamental question in immunology. How does the body discriminate self from non-self, and how does this process turn on the immune response? So we've set up a pretty effective pipeline, if you will, to study virus infection in the lab. The pipeline starts with people in the lab doing work at the lab bench. It's very similar to working at your countertop in your kitchen. We get things out and we mix them together, and then we see what happens. And then in addition to that, what you don't do usually in your kitchen, you don't take your food and put it through a computer. You know, some people can do that now, but in the lab, we actually take what we produce in the lab and we break it down to molecular elements, if you will, and run it through processes that we call next generation sequencing for high throughput sequencing of the nucleic acid that's produced inside of a cell. And that is fed into the computer. Then we have a bunch of bioinformaticists who take that information and build gene expression profiles and tell us what might be happening inside the cell. And then we build models to test hypotheses. And in the end, we feed everything back into the computer, and the computer gets a little bit smarter each time we do this, and it'll tell us what's going on. And you can see this network here. In red shows a molecule called RIG-I, which we discovered in part several years ago, is now one of the headliners of what I'm going to tell you about, innate immunity. So this is our pipeline. We use it every day to study infection and immunity, infection induced by viruses. The lab mostly studies viruses that are called RNA viruses. They have an RNA genome, as opposed to DNA viruses, which have a DNA genome. So RNA viruses are influenza, hepatitis C, Zika, things you're familiar with. DNA viruses would be, for example, herpes virus, smallpox virus. They have DNA genomes. So fundamentally different, but they're all viruses. So when a virus infects us, a lot of things happen right away. At the site of infection, there's a lot of action. And then that action is meant to develop a systemic immune response. So I'm going to talk later about West Nile virus and Zika, and these are viruses that are transmitted by mosquito. So if you're unlucky enough to get bit by an infected mosquito, you could contract West Nile or Zika virus infection. So if a mosquito bites me here, at the site of infection, a lot of stuff is going to take place like you see in this figure. And underneath my skin are actually immune cells that are called innate immune cells that are surveying my body for infection and for other bad stuff. So when the virus gets deposited there, there's going to be an area of inflammation, and the cells under here are actually going to grab the virus and they're going to traffic it to the draining lymph node, which is going to be here in my armpit. But what happens at the site of infection is the virus is recognized as non-self, and at that site of infection, which could be here where the mosquito bit me, the immune response is going to start right there at that site inside perhaps the single infected cell that received the virus. And inside that cell, there's going to be a remarkable change. A bunch of genes are going to be turned on. You see here in red. Each line horizontally is a gene. And the genes are getting turned on after West Nile virus infection, after the virus, say, bites me right here. And a bunch of other genes in green are getting turned off. So the virus is becoming an antiviral factory the cell's becoming an antiviral factory to try to fight off the virus. If that doesn't work, the virus is going to spread, and the immune response systemically is going to be geared up because now those cells that were hiding under my skin are carrying pieces of the virus to my lymph node. So the lymph node under my arm, for example, is going to be engaged with viral pieces. And those cells will present those viral pieces to my other immune cells, and then the immune response is going to take off. And over a period of a week, I'm going to have a massive expansion of the lymphocytes in my body towards fighting off this virus, but not before the virus has been recognized by as non-self. If none of the recognition happens, none of these events will take place. And we know enough that we've modeled this in mice, for example. If the recognition doesn't take place, the mice are highly susceptible and they're going to die from infection. And you should know that mice have an immune response very similar to ours, an immune system that's shared by humans. So they're a good model for testing these things. So all the stuff I'm going to tell you about is happening up here at the very first engagement of virus infection. Viruses are obligate intracellular parasites. So when they infect you, they can't live without you. They have to parasitize the metabolic processes in your cell in order to replicate, make new virus, and spread. So a virus includes one or more pieces of nucleic acid. It can be a single piece or 2 pieces or more. Flu is actually 8 pieces of nucleic acid. And then it's surrounded by a capsid, a proteinaceous shell we call it. And some viruses even have a lipid envelope around them, not all of them. And then on top of that lipid envelope, they might have molecules called proteins or co-receptors that bind to the target cell and mediate the infection. So what we know is that these different pieces of the virus are recognized by the body as pathogen-associated molecular patterns. We call them PAMPs. So we talk about PAMPs all day long, but the body has to recognize the PAMP in order to start the immune response. And we have a lot of effort going on to understand what is a PAMP. And what we've learned is that PAMPs are often viral nucleic acids, viral RNA or viral DNA. In this case, I'm going to talk about viral RNA. And I'm going to walk you through now a generalized replication cycle of the RNA viruses that I'll be talking about. So what I'm going to show you is how hepatitis C replicates, how Zika replicates, and so on. So the virus first, Zika would be deposited into my skin by the bite of a mosquito. I might receive hepatitis C through blood contamination or contamination from a contaminated needle or something like that was introduced into my blood. But nonetheless, however that virus comes in, they can come in through multiple routes. It has to be recognized as non-self in order to kick off the immune response. So viruses enter the cell by binding to receptors, and then they're brought into the cell through a variety of mechanisms. one thing they all have to do is they all have to uncoat. All that capsid in the envelope I showed you has to be pulled away from the nucleic acid, and the nucleic acid has to be liberated so that now it can facilitate virus replication. The virus has to make what we call a messenger RNA. That's what this is. And then the next thing it does is it makes proteins. And so when you get infected, before you even know you're infected, your cell is pumping out viral proteins within the first 24 to 48 hours of infection. And when those proteins are made, then the viral genome starts to replicate. This is when you're really in trouble, because when that viral genome replicates, it's going to make more viruses. And then the viral proteins all come together. And like the virus I showed you earlier, new viruses are made, and they're released from the cell through a variety of ways. So now when this happens, you're going to have virus floating around your blood, infecting all kinds of other cells. And what your immune response wants to do is stop this process dead in its tracks so we don't get sick. And most of the time, we do this. On a daily basis, we're infected over and over again, but your innate immune response that I'm going to tell you about protects you. When you get sick and symptomatic, the virus has found a way around your innate immune response and your immune response against infection. So these arrows point to sites that are in the virus replication cycle where nucleic acid accumulates or is exposed to the inside of the cell. And we know that's what kicks off the immune response. I'll talk about that more in a moment, but when that happens, it happens within minutes or hours after infection. It's a good thing because that's what kicks off immunity. This is very different from other parts of the immune response that take minutes or hours or days to start. And then the adaptive immune response, where you have antibodies and cells that kill infected cells, that literally takes days or weeks. So innate immunity happens like that, as fast, sometimes as fast as we can measure it in the lab. And this is what protects all of us on a daily basis from getting sick, just in general life. So in the end, innate immunity, what we've learned, is more than this. It seems to program the entire immune response. Without innate immunity, we can still get an immune response, but it's totally weak and totally messed up, and it's not protective if we actually survive that far. So what we try to do is learn everything we can, and then our goal is to build therapeutics or vaccines that can help, that have maybe a public health impact. Years ago, we identified a molecule called RIG-I that stands for retinoic acid inducible gene I. RIG-I happens to be the sensor that tells your body it's infected. It has a close cousin called MDA5 and another distantly related cousin called LGP2. But these are molecules that are expressed in all of the cells in your body, and they are sentinels that will detect the presence of an RNA virus infection, the presence of flu, the presence of measles, the presence of Zika or hepatitis C will be detected by RIG-I or MDA5. And they work by interacting with this molecule called MAVs. And when they do that, there's a reaction in your cell that turns on all the genes that I showed you to fight off virus infection. But what's interesting here is these molecules are pathogen recognition receptors. So they'll actually recognize non-self nucleic acid that's present in your cells. And thank goodness, because when they do that, they detect the presence of a virus that could otherwise kill you. And we know that they recognize very special nucleic acids that are marked as non-self. Typically, we don't have double-stranded RNA present in our cells at high abundance. When this shows up, RIG-I or MDA5 can find it and kick off the immune response. Typically, we don't have single-stranded RNA that has, the presence of a 5' triphosphate, for example, marked. The RNA is marked with things that are different from self. So RIG-I can see this and respond to the RNA and tell your body it's infected. And typically, we don't have big, bulky adducts of loopy RNA hanging around in our cells. When we do, we know it's a virus, and RIG-I or MDA5 will recognize it and turn on the immune response. So what happens in this cartoon, here's a virus infecting a cell. So it could be a a cell here in my skin, and then a virus infecting a cell in my skin, and then inside that cell, RIG-I is sensing the viral RNA. And you can see that we've drawn it in here. It's one of my teaching slides. And then RIG-I gets activated. It binds to MAVS protein, and then it signals downstream to turn on things called transcription factors. But what happens, we can measure that in the lab because this is a transcription factor that goes into the nucleus and turns on genes that fight off the virus. And you can see this is the IRF3 factor. You see that here in the cartoon. In cells that aren't infected, it's all outside the nucleus. And then look what happens after virus infection. It shuttles right into the nucleus, and that turns on the immune response. RIG-I does this. So when the virus goes in the cell, RIG-I recognizes it, and it sends IRF3 and other stuff to the nucleus, and that turns on genes that fight off the infection. We measure this in the lab, and we know this is a very robust process that happens literally within minutes after virus infection. And then when this happens, your cells start pumping out all kinds of stuff. And one of the things that pumps out in great abundance is called interferon. And so if you've ever had, who's had the flu in here, the real flu, where you're bedridden, okay. So the reason you're bedridden is because your body's making so much of this interferon and it's flowing through your blood. The interferon binds to interferon receptors in all the cells of your body, and it turns on the expression of literally hundreds or maybe thousands of new genes that never get turned on unless you're infected. And when that happens, it gives you the chills, it gives you a fever, it makes you feel really bad, and it's your body fighting off the infection, which is a good thing, actually puts you to bed for a week when you have flu. Flu, pathology of contemporary flu, for most healthy people is all about this massive interferon response that's there to fight off the virus. More to it than that, but interferon is a very potent antiviral mediator, and it has some side effects with that protection. In addition, your cell becomes, all the cells in your body become better able to tell the rest of the body, I'm infected. So they put pieces of the virus outside the cell and kind of wave it around so the immune response can find it. And then, like I was telling you about in my draining lymph node, a bunch of immune cells will traffic out of my blood and go to this lymph node to get educated about the virus. And your lymph nodes swell up. That's how you know this is going on. And they're sore and you can feel it. But that's also a good thing because all those immune cells are in there getting trained to fight off the infection. This is what innate immunity does, and it's very fast. So it all starts with a molecule that we identified, and my colleague in Japan identified about the same time. This is called retinoic acid inducible gene I, so we call it RIG-I. And we solved the structure of it with some other colleagues, and it looks like this. This is a classical crystal structure of a molecule. What it shows you are some interesting things. Embedded in here is when we crystallize it, there was a PAMP RNA already in there. So RIG-I was recognizing something. There's an RNA. And then this red part that we call a repressor domain makes first contact. So RIG-I is a first responder. And the red part, the repressor domain, actually can sense the nucleic acid and make an interaction with it. And then when that happens, RIG-I undergoes a change, and it wraps around the nucleic acid like a barrel, like you see here. And then the immune response can start because RIG-I undergoes a conformation change where now a part of the molecule is sticking out like my thumb inside the cell. And that part of the molecule called the card can bind to other cards of other proteins. And this facilitates what we call signal transduction. Tells the body I'm infected and we need to start an immune response. The RIG-I cards bind to a molecule called MAVS. So it was identified by Ming Lu in the lab about 2005. So what happens is when RIG-I undergoes a change and it can bind to MAVS, then MAVS recruits all these other molecules, and there's IRF3 again. Remember I told you it goes into the nucleus. It interacts with itself and goes off into the nucleus and turns on gene expression. This process happens within minutes or hours after infection. And we first learned about RIG-I when we were studying hepatitis C. Hepatitis C had a global prevalence, nearly 200 million people. and not widely known because it never got a lot of press. But hepatitis C virus, in the worst case scenario, can destroy your liver and kill you. It's a major cause of liver cancer, and is distributed worldwide. So basically, when the guys came home from World War II, some of them somehow got exposed to a new virus. Didn't know what it was. It turns out it was hepatitis C, and they brought it home with them. And it ended up getting into the blood supply, and it spread worldwide. And only recently do we have an effective antiviral therapeutic. But let me tell you something about hepatitis C, because it infects your liver, and you can see this nice healthy liver right here can get really bad and become cirrhotic in the worst case scenario, hepatitis C. And this one has a bunch of cancer nodules all over it, showing you how bad it can really get. So nearly 2% of the world has chronic hepatitis C infection. We can treat it now. The worst case scenario is that the virus can track with HIV. So it's a #1 killer in America and Europe of people that have HIV infection. Hepatitis C is what kills them today, not the HIV, because they can get treated for the HIV. So it's still a major problem that we're working on. And this is kind of what it looks like in cartoon form. My daughter, Tennille, who's here tonight, made this for me when she was really little. And I use it all the time now. So hepatitis C is a virus with an envelope, like I showed you earlier. And it gets into the blood. And when it gets into the blood, it makes its way to the liver, infects the liver, and that's when the problems start. So using our pipeline that I told you, we took a look at hepatitis C liver, pieces of liver from hepatitis C infected patients. And we know that when genes get turned on in our computer analysis, they'll show up as red. If they get turned off, they show up as green. And what we expect is when we get infected with a virus, all these genes, these are all innate immune genes I'm showing you, this whole figure should be blazing red, but you can see it's only partially red. And we realized right away that hepatitis C was different. It seemed to be blocking the innate immune response. We didn't know how. We didn't know even how the innate immune response could get turned on. We just knew it gets turned on. But this was our first clue that this virus could manipulate innate immunity, and it might be bad for the outcome of hepatitis C infection. So what have we learned since then? This is a cartoon of the hepatitis C genome. And each line across this color spectrum is a different protein. So when you get infected with hepatitis C, it makes one gigantic protein, and then it cleaves the protein into 10 little proteins, and they all go to work doing their job replicating virus, but they are also a cause of disease. And what we know is that hepatitis C gets into the cell and it sets up shop and it starts to replicate. Each of these little dots is a replication factory inside the infected liver cell. So they're just different colors because we're looking at two different proteins, and when you put them together, the red and the green makes yellow, so we know those are working together in a complex to replicate the virus. This is bad because we don't want to replicate the virus. So what we know is that the body senses hepatitis C by virtue of RIG-I, and the hepatitis C studies led us to RIG-I, and they led us to MAVs and the whole innate immune pathway that we now understand is very important for recognizing most RNA viruses, if not all of them. Flu, measles, all the stuff you know about, probably recognized through this pathway to tell the body it's infected. So what's more is there's a little part of the nucleic acid of the hepatitis C virus at the very end here that encodes a region that we call the PAMP, the pathogen-associated molecular pattern. That PAMP we nicknamed it the poly-UUC region. That's not real important except to know that it works in cooperation with a couple other things in the viral genome to tell the body that, hey, you're infected. RIG-I recognizes that, binds to the viral genome itself, undergoes this conformation change so it can signal, like I showed you, and then the immune response starts. So it was this piece of nucleic acid that really told us how viruses are recognized. And this work goes back about 10 years now, been worked on by several people in the group. In the liver, then when RIG-I recognizes the virus, remember that molecule IRF3 that I showed you? It should go to the nucleus. If it doesn't go to the nucleus, there's something wrong. So we know that in acute hepatitis C virus infection, IRF3 does go to the nucleus. That's what you're looking at in red here, okay? Blue is the cell nucleus. You can see they kind of go to the same place. So this is good. But in chronic infection, look at the red. None of it's in the nucleus. The blue is by itself, and there's no red over top of it. And in fact, the green here shows the cells that have hepatitis C replicating in them in this human liver, and IRF3 should be in the nucleus because RIG-I should recognize it and send, RIG-I should recognize the virus and send IRF3 to the nucleus, but it's not there. So it was this experiment that told us there's something really bad going on here. The virus must be blocking the innate immune response. Well, so this arrow points to where we think the blockade happens in the pathway. We figured out it must be happening at a level below RIG-I, but probably not too far away from it. And it turns out that Ming Lu discovered a molecule that was later named MAVS that gets cut by the virus. The virus encodes its own protease. This is a protein that cuts up other proteins. And it evolved to have its own protease to target the MAVS in our body, cut it so it's inactive, and knock out the entire RIG-I pathway. And it does this within 12 hours after infection. So once you get exposed to hepatitis C, in 12 hours, it's over. The virus wins. It's the most efficient virus at causing a chronic lifelong infection that I've ever seen. So if you take a look at this, this is called an immunoblot assay. And Ming is looking at MAVS. Here's the molecule, what it looks like in our body now. Here's what it looks like when you're infected with hepatitis C. You'll see it's a little bit different. It's a little bit smaller. We call this smaller. It has a lower molecular weight because hepatitis C has chopped a piece of it off. And when it does this, the whole RIG-I pathway is dead. It's an incredible story of viral evolution to render our innate immune defenses harmless to it so it can replicate and spread. And what Ming found out was that there's a sequence inside of MAVS that looks like this, and this happens to be the same sequence that's in the viral protein itself. So the protease actually chops up the viral polyprotein at these sites to liberate smaller proteins, and it also chops MAVS because MAVS has that same sequence. This virus evolved this sequence, probably coevolution with the human protein to chop MAVS so it could lead to a persistent infection. The virus wants to persist, so it can make a lot more virus and spread them throughout the humans and keep living. And hepatitis C does a pretty good job of this because it's silent for many years, and then after a while your liver can't take it anymore. But you're like 50, 60 years old, but you've had it for 30 years. And the virus is very successful because during those 30 years, you would have had many opportunities to transmit the virus to other people, sexually and through transmission of blood-related products. If you look in the cell, the MAVS protein is a beautiful mitochondria look. This is what mitochondria look like in your cell. Mitochondria is the energy factory inside your cell. But this is what they look like. And then MAVS is planted on the mitochondria. When it gets targeted and cleaved by hepatitis C, it floats off the mitochondria and it looks like this. There's plenty of MAVS around, but it's no longer on the mitochondria. It has to be on the mitochondria to signal. So this experiment was also done by Ming Lu, and it was a groundbreaking experiment because this told us that a couple of things. The virus cleaves MAVS and renders a RIG-I pathway useless, but MAVS has to be on the mitochondria to signal. And since then, we and many other groups have learned that the mitochondria membrane is a very important signaling platform, not just for RIG-I, but all kinds of stuff that causes inflammation and very prevalent in autoimmune disease. So this was a milestone for us. And the MAVS is cleaved by the virus through the actions of what's called the NS3 protease. It looks like this. Not dissimilar from the look of rig eye, but this is what the hepatitis C protease looks like. And we knew, and many other people knew, that we could make small molecules that would bind to the protease and prevent it from chopping up other proteins. This is what one of the protease inhibitors look like that we started working with a company called Intermune. So if you consider hepatitis C virus infection and MAVs, in a normal cell, MAVs looks like this, nice mitochondria look. In a hepatitis C infected cell, it looks like that. It's very bad because the RIG-I pathway is not going to work. It won't protect you. But if you treat with the protease inhibitor, look what happens. MAV starts to come back. The virus doesn't replicate anymore because you've blocked the protease and you've restored the innate immune response. And when we figured this out, it was a terrific discovery because we realized right away that the protease inhibitor drugs would be able to restore the innate immune response and help facilitate the clearance of hepatitis C virus. So a person now with a chronic infection who could never be cured, or maybe only half people could be cured with a very, very unpleasant therapy, can now be cured completely with the protease inhibitor and new generation molecules that go with it. So what we've learned is if we apply the protease inhibitor to an infected person, whereas this blockade was rendered by hepatitis C virus infection, now this whole pathway can be restored and the pathway can operate to clear out the virus. It works very well. We can cure most infections today. And on top of that, when the virus is removed from the liver, then the liver can start working normally. And if we get infected with something else, the liver can clear it out. So we're pretty pleased with this outcome. But it's not over yet. Hepatitis C, excuse me, is still a big problem. The direct-acting antivirals, they come in a pill. You have to take between 24 and 84 pills. It's $1,000 a pill, very expensive. Developing countries can't afford it, but the drug companies haven't dropped their prices for them. The people who are cured can still get infected again because there's no immune protection. The pills don't protect you from reinfection. They just clear out your existing infection. The other thing is that hepatitis C virus still remains a major source of liver cancer. And what we really need is an effective vaccine to go with the antiviral treatment. So we're not out of the woods yet with hepatitis C, but we're getting there. Okay, so that's some of the hepatitis C story. We're still working on it. We have several people in the lab working on hepatitis C, trying to understand the molecular basis of liver inflammation, the fundamental aspect of hepatitis. The other thing that we work on are viruses that related to hepatitis C. And so this slide just shows the other viruses that are called flaviviruses. Hepatitis C is a member of the flavivirus, Flaviviridae family. And other viruses related to it include Zika, West Nile virus, yellow fever, and dengue virus. Many of these are tropical diseases that are now showing up at our doorstep. Our experience with hepatitis C taught us a lot about these type of viruses, but it didn't teach us everything because these other viruses are transmitted by mosquito. Hepatitis C isn't. When West Nile virus showed up in the United States in 1999, we actually recovered the virus from the brain of a dead grackle, a big blackbird in Texas. One of my students brought this dead bird to the lab on his way to work. He found it in his yard, a guy named Brian Keller, and we actually took it and we ground up the parts and we recovered West Nile virus from it. West Nile virus Texas, it's called, is now the dominant West Nile virus strain that's circulating in the US. West Nile virus looks kind of like hepatitis C, and what you need to know is it's transmitted by mosquito. So if you go into endemic areas, you want to consider some kind of protection against mosquitoes. The other thing is West Nile virus is here in Washington State, and when I came here in 2007, I had to meet with the biosafety committee at the University of Washington who were very concerned because somebody in Shelton had just died of West Nile virus infection. So we had this big talk about the type of biocontainment we need for it. And eventually we got USDA permission and Washington Public Health permission to bring West Nile virus to the University of Washington. I think we were the first people to do that. So we have a West Nile virus research program in my lab. West Nile virus is distributed from east to west. It came to the United States around 1999 to New York City. And then because it infects mosquitoes and mosquitoes bite birds, the birds fly. The major flight routes of birds are shown here. Every bird, did you know every bird in America, every bird in North America goes to Texas on its way somewhere. And they stop in Dallas, Texas, like you wouldn't believe. And then they go to the southern coast of Texas, and then they disseminate to South America, wherever they go. But what happened is that Texas got hit really hard by West Nile virus. It hit in 2000, 2002, and then it came back with a vengeance in 2012, 58,000 cases with a case fatality rate of 9.8%. So that's up from 5% the last time it showed up. So that could happen here. So we need to be prepared. The thing about West Nile virus is it's a neurotropic virus. So bites me here, the mosquito bites me, the virus is going to go to my spleen, replicate a bunch of times, and then it's going to end up in my brain, and that's when things get really bad. Zika is very similar. It's spread by a mosquito. It emerged recently from Oceania across the Pacific to South America. It's working its way up north. It's transmitted by Aedes aegypti, which is a different mosquito. So the good thing about Aedes aegypti is it can't survive very well in the colder climate of North America, but it's probably present in Southern California, southern United States, all the way to southern Florida, where they actually have had de novo Zika cases. So we're very concerned about it because what happened with West Nile virus is it came in and only infected one species of Culex mosquito, and then it became transmitted to birds, and then Anopheles mosquitoes, a different species, fed on the birds, and West Nile got into the Anopheles mosquito, and then it got into subspecies of all those mosquitoes, and now it's in like 12 different mosquito subspecies across the United States. So if that happens with Zika, we're going to have a major virus dissemination problem on our hands. Right now, that hasn't happened, but the lesson from West Nile virus tells me it can happen. So Zika could be a threat to the USA. There have been cases here, but they've been travelers' cases. And soon we'll have actually mosquito-transmitted cases, and that's not going to be a good thing because Zika presents a whole nother challenge when we think about maternal-fetal transmission of the virus. The other thing you want to know is that Zika virus can be transmitted not just from the bite of a mosquito, but sexually. So that's very different from the other flaviviruses. And in addition to that, whereas West Nile virus can't be transmitted from human to human, Zika virus can, from a mosquito biting me and biting any of you if I have Zika, and also through sexual transmission of blood products. So Zika is a special problem. We haven't seen anything like this in the US, but it's coming. So we want to be ready. So what we've done at the University of Washington is we've set up a program for maternal-fetal health against virus infection with my colleague Christina Adams-Waldorf, who's here tonight. And we're happy to tell you that we just got a couple of new grants to fund this program, so we're pretty excited. And this is Christina teaching me about placenta. But we have a group of folks here that we all work together as a team to study Zika virus. Zika does a lot of things, but the thing that it's most famous for is causing microcephaly. And this is a very bad outcome because it's transmitted in utero from mother to fetus. The fetus gets infected. Zika loves fetal neuroprogenitor cells, and we don't know exactly why yet, but we're going to figure it out. And when it infects those cells, it literally destroys the normal architecture of the brain, so the brain doesn't develop, and the skull follows the development of the brain, so you get the appearance of the microcephalic child, but the outcome is way worse than it looks even. So it's totally debilitating to the child, and it has a major impact, as you might guess, on the family. So Zika loves neuroprogenitor cells, and you don't need to try to read this, but the point I'm trying to show you here is that there's a normal architecture of the brain, and when Zika infects the fetal brain, that architecture, which might look like this, now looks like this, and it's a total disorganized array. What we know is we can grow the flaviviruses, West Nile and Zika, in the lab, and you can look at this image and see IRF3 in the nucleus. That's what the red is. This is nuclear IRF3. So Zika turns on the RIG-I pathway. West Nile turns on the RIG-I pathway. The white is our detection of the PAMP that turns on the RIG-I pathway. So the cells are producing the PAMP, RIG-I is recognizing it and sending IR3 to the nucleus. So these cells now are going to try to fight off the virus infection. They should fight it off, but why don't they? Why do people get sick with Zika? Well, so what we know is that this left side of the pathway, the RIG-I pathway, is activated with the Zika infection. So it should control the infection, but it doesn't. So we took a look at the right side. This is the interferon side, the side that I said makes you feel sick when you have the flu. It's a very important aspect of immunity because we need interferon to turn on hundreds of genes to fight off the infection. So we can look at the interferon pathway because we study molecules called STATs. And STATs get turned on, you can see that with this dark blob here, after interferon is bathing the cell during a virus infection. But look what happens in a West Nile infection. This molecule, STAT1, doesn't get turned on, okay? So now if you look further, there's a whole family of STATs, and none of them get turned on. Compare this to this, this to this, and so on. No STAT activation. That's a big deal. We've never seen virus that wipes out all of the STATs. Maybe they might wipe out one of the STATs, but all of them, that's crazy. So West Nile virus shuts down all the STATs. So you can have the best RIG-I ever, but if the right side of the pathway doesn't work, you're in big trouble. That's what these viruses do. Now take a look at Zika. It's the same thing. Zika and West Nile virus put this broad blockade on this it's called the JAK-STAT pathway, so interferon doesn't work. So you can turn on the RIG-I business all you want, but the virus doesn't care because it blocks the downstream actions of that pathway. And what's more remarkable, if you look here, take a look at this. So the red shows STAT in the nucleus, as it should be when it turns into the blob like you see here. But this cell that's infected, there's a little bit of Zika right there, totally blocks it. So if Zika gets in the cell, your STATs aren't going to work. And this is remarkable for a variety of reasons because you have an attenuated innate immune response, but think about the outcome of Zika in a fetal development, microcephalies, if the fetus survives. But you know the brain development is programmed through STAT signaling, that as we grow in utero, the cells of the developing fetus produce cytokines that direct the development of the brain. So when those cytokines get produced as the fetus develops, they tell the cells of the brain to make neurons, to make glial cells, to make oligodendrocytes. And when all those are made, then your brain develops and you have a nice, beautiful head and a beautiful baby. But what happens in Zika virus infection is the STATs are blocked. I just showed you this broad STAT blockade. It's crazy. Now, what we've done here, and this is work done by Justin Roby in the lab. It's pretty new stuff. But these are some of the brain cytokines, they're called, that turn on the STATs to make the brain grow the right way. And when we treat neuroprogenitor cells with them in a cell culture dish, it actually turns on the STAT. This is called STAT3. You see that big blob? That's good. And look what happens in the presence of Zika. These are actual neuroprogenitor cells that would get infected in the fetus. Zika blocks the turning on the stat. And because of that, we have less development of those cells into neurons or into glial cells. And if you take a look at a picture, it looks like this. So up here, these are the cells without Zika, turning into neurons and turning into what are called astrocytes, beautiful brain cells. And in the presence of Zika, you can see how the image is not as bright because there's fewer of those cells differentiating into neurons and astrocytes. So you put that in the context of a fetal brain, the brain isn't going to develop, and you might end up likely with microcephaly or worse, inability to survive gestation. So what we think is happening, and this is a hypothesis still, we have to formally prove it, but we think that Zika blocks the stats and this blocks brain development. This could be the molecular basis of microcephaly. So think about that. If we could find a way to prevent this from happening once we understand that mother may be infected with Zika virus while her fetus is growing, if we had a way to disrupt this process, we could save the baby from neurogenic pathology. So that's one of the big goals in the lab. And you can see here, now with Zika and West Nile, we can draw an X to the right side of the pathway. Hepatitis C messed with the left side of the pathway. The take-home message is viruses mess with every pathway you can think of that controls the immune response. So if you want to study the immunity, you should study pathogens, because they'll tell you everything that's going on. I was just in Salvador, Brazil with my wife, and we were meeting with some Zika researcher colleagues of ours, but they took us to Zika clinics, and it was mind-blowing to me because here in the US, some of the public health messages we're getting is that Zika is no longer a problem. It's like, subsided. But I have to tell you, in Brazil, it's a huge problem. They had a wave of Zika that came out, and they have now to set up special microcephaly clinics around the city of Salvador and elsewhere to handle treatment and rehabilitation of the Zika children. And this is just some of them that we saw. It's very devastating. We visited a couple of clinics. They also set up schools, special schools for the Zika kids. This is one of them we went to. It's my wife sitting there talking to some of the kids that have the least debilitation, but these are Zika kids. The worst ones are the microcephaly kids. These kids are very special. They're happy. Important thing is they're happy, but they're dealing with cognitive disorders from a previous Zika infection. The other remarkable thing was that they even have Zika dental clinics because the Zika kids have to have special handling just to see the dentist, for example. It's a huge problem. So Zika's just starting. We're worried that it's going to come north, going to come here. There's no approved vaccine. There's vaccines in the pipeline. They have to be tested. It's going to take a while to get them approved. And then even if they get approved, it's not clear they'll ever get produced in massive quantities and disseminated. So that's a public health discussion. There's no antiviral drugs, like the drugs for hepatitis C. We do think some of the hepatitis C drugs, though, might work against Zika. We're currently testing that with Christina. Mosquito spread, I talked about that. Zika's probably going to spread to different mosquito species. And then, of course, we need aggressive research to identify targets for therapeutic intervention. Lessons from viruses are many. And the lesson that we've learned is the RIG-I pathway is very important for viruses, but viruses already know this, so they try to remove the RIG-I pathway as a threat. We think we can beat them, though, because we can tackle them by leveraging RIG-I and its cousins as therapeutic targets. That's one thing we've learned. So what we've done is we put together a group of people, small biotech companies and others that work with us, to ask if we can make drugs that could turn on RIG-I and fight off virus infection. So our strategy really going into this was we knew what rig eye looked like because we have the molecular structure of it right here. The red part recognizes the PAMP. The red part close up looks like this. It's got there's a little ditch there you're looking at. It's called a ligand binding cleft. That's where the PAMP lies inside that groove. And then those codes that you see there, those are amino acids. And when the medicinal chemists looked at this, they got really excited. They said, this is a perfect drug binding cleft. We can do this. So we put together a team and we started screening small molecules. We identified some that could bind in this cleft. The idea is they could turn on rig eye and fight off the virus infection by turning on all of our innate immune defenses. So that works. I'm not going to walk you through the data, but it actually works. We've seen efficacy against a broad range of viruses. Published this recently, everything from Ebola, dengue virus, coronaviruses, and flu can be tackled, we think, with rig eye therapeutic. So think about this. If you have a drug that targets a virus, eventually the virus is going to mutate and the drug won't be effective. We learned that from HIV. But if you have a drug that targets a host, and if the host molecule turns on hundreds of genes, that's like hitting the virus with hundreds of drugs. There's no way the virus is going to resist that. What we have to be careful to control is toxicity, because you don't want to be in bed like the flu scenario when the innate immune response puts you to bed. So we're working on this rig eye program to produce small molecules that can be used as drugs. Those molecules are of interest to pharmaceutical companies. We just licensed some of them to Pfizer. And so Pfizer wants to develop these as immune agonists to enhance the immune response against cancer, infection, and all kinds of stuff. So that could be a success story, but we'll see. Rig eye targeting, we think, is very attractive. And that's pretty much the end of my talk. I want to summarize just by reiterating through this cartoon the rig eye pathway. It's very important for daily life and everyday health. Protects us on a daily basis. And if it doesn't, you're going to get sick. And those points are made here. I told you about hepatitis C, West Nile and Zika, and how we think we can leverage this pathway for therapeutics. The last thing I want to tell you about as I close is the Center for Innate Immunity and Immune Disease, which we started here a few years ago. My colleagues Ed Clark and Keith Elkin are here as co-directors. We set up this center to help scientists in Seattle and elsewhere do innate immunity research, which has gotten really big. Innate immunity has come to the forefront. Innate immunity is important for cancer, autoimmunity, infectious disease, vaccines, like I said, everyday life. Part of the center, though, has an outreach, and our mission at the University of Washington is to train and teach. We want to train the next generation of scientists. The Center for Innate Immunity and Immune Disease has an outreach program. And so you can see here's our U.S. Senator and the Director of the National Institutes of Health visiting this kid. He did pretty well. You see the Dean of Medicine's back here, Paul Ramsey, a few other people. And he did pretty well answering questions. They go, son, that's good. Where do you want to go to college? And he said, well, I think University of California. But so, I was going to say University of Washington. But anyway, the outreach program has been successful. So these kids, they gown up in all of our gear to see what it's like, at least wearing it to handle pathogens, to work in a lab, do experiments, looking at the microscope. The kids these days are so talented, I can't tell you how impressive they are. But the outreach program still needs help. We're looking for people to help us and keep everything rolling. So the last message I want to leave you with is doing big science is not easy. We're constantly begging for money, and most of the time we fail at our experiments. 90% of the time we get negative results. So the scientists in the crowd, they already know this, like preaching to the choir, but it's not, every day is not like the discovery of rig eye or discovery of MABs, cleavage by hepatitis C day. Every day is like, oh man, why didn't that work? And being a scientist takes a special person. You get rejected, you do your best work, you write a science report, submit it to a medical journal, and your peers review it and they reject it. Oh, this is no good. When we discovered Rig-I, it didn't even warrant enough attention to get in one of the high-impact medical journals. Since then, it's been cited over 1,000 times. And now everybody realizes that was probably pretty important. But that's daily life for a scientist. It's a lot of rejection, and you have to really have a thick skin and be resilient to all of these rejections. It's the discoveries, even the small ones, that keep us going. So I want to leave you with that. And just final thought is to talk about my mentors a little bit. That's how I got here. And my goal in life is to mentor others. And I got my bachelor's degree and was lucky enough to get hired by Ed Clark, who's here tonight. And he put me on this project on this new virus that was just discovered. It's called HIV. And we set up some of the first infection models to actually show that HIV can cause AIDS. So that was pretty exciting for a young guy just out of college. And then I got interested in pursuing the education further, and the new graduate program just started that was really focused on human disease, very attractive to a young guy also. George Kinney designed the program. He's not here tonight, couldn't make it, but I want to acknowledge George for developing the Pathobiology graduate program, which is a front-ranked program that attracts international students today. Marilyn Parsons was my mentor in graduate school. She's here tonight. And, you know, she brought me into the lab and taught me molecular biology. Here she is showing how to streak a bacteria on a plate. And, you know, we grow DNA in bacteria to do things with it. So that was very important. And she mentored me, and we cloned, discovered the first protein kinase from a protozoa. We were studying African trypanosomes that cause African sleeping sickness and figured out that they grow because of molecules called protein kinases. That's what got me interested in signal transduction. And since then, I was mentored by several people who, many are here tonight. It's my pleasure to be part of the same faculty that actually taught me. I'm in the graduate faculty of pathobiology, and I work with these people. So I want to thank all of them and all of you for coming. And that's all I have to say. Thanks a lot.
Okay, so we have time for a couple of questions. I have a question to ask, but I'm going to see whether or not we have someone ready and raring to go in terms of questions. Yes, no? Up in the back? Okay.
So thank you, Dr. Gale. It was very interesting. I'm not a prophet, but I'm guessing that the general public and politicians and policymakers don't understand a lot of the practical issues in medical research. If you had a magic wand and could knock out 2 or 3 of those things they don't understand, what would those be, and more importantly, why?
The biggest one, the biggest hurdle for us at any given time is funding. So how do you control, how do you facilitate funding for certain programs? And I can't think of a better system than what we have in the United States, but it's still not perfect. And a lot of times we feel the peer review could be improved, and the government supporting the National Institutes of Health budget-wise is a big issue for us. Every time a new president comes around, we just shake in our boots. What's going to happen to NIH budget? And what happened recently was that the NIH, what we enjoy is that NIH has bipartisan support. So when a sitting president says he or she is going to reduce the NIH budget, the lawmakers in our country stand up and come to our aid, and we've always appreciated that. I'm worried, though, with what's happening in Washington, that's not always going to happen now. So that's the one thing I would wave my magic wand at and get back to funding levels that are like 20 to 22%. There was a time in the recent past we had dropped down to 6% success rate. We can't compete. It's an issue of national security. China funds their research at 40 to 50% success. And if you want to compete on a global scale, you have to meet the challenge.
Anyone else?
Yeah, this is a public health question. The Zika virus, I'm wondering if there's, at what stage in child development does the Zika virus stop infecting the cells? We have to worry about 6-month-old kids out on the playgrounds getting infected with a mosquito, and having that infection with the Zika virus affect their development even after birth.
Yeah, that's a great question. What we understand today is anybody at any age at any time is susceptible to Zika virus infection. So it's a real danger. The virus in a young child, are young children more vulnerable to the virus? We know what the fetus is. As we progress in age, there's not enough studies that have been done to know if there is differential susceptibility with age. But the bottom line is that at any time, anybody's susceptible to Zika. And so a child on a playground would be a concern. So, yeah.
Looking out in the audience, any other questions? So I'm actually surprised that none of them have asked this, so I'm going to ask you, because often, I know as a public health person, we talk about these problems and we're so used to talking about them that we forget that when people hear about some of this stuff for the first time, that they get a little freaked out. So I think probably in the back of the minds of a lot of people out there is, what can I do to protect myself and my family from these types of infections? What would you recommend?
Well, first of all, I don't want people to be so afraid of Zika that you're not going to go hiking and go outdoors. So you can pay attention to what the public health experts identify as Zika endemic areas. And if you're pregnant, you may not want to visit those areas. If you're a young man and want to have a family, you may not want to visit those areas. If it turns out that Seattle becomes a Zika endemic area, then we need to take precautions like mosquito repellent and long sleeves instead of short sleeves and things like that. Hepatitis C, is a different set of precautions. Don't share contaminated needles. It's highly transmissible through IV drug use. It's a main high risk population with hepatitis C. If you are concerned about HIV, protected sex is very important, and know your partner and those kind of things. So public health is also all about public good sense. So you don't do risky things if you know they're risky, and that'll reduce your risk. And that's easier said than done because viruses are sneaky. And so you want to be self-aware, know where endemic areas are, and know that in developing countries, blood can be contaminated. So if you travel there and have an accident, in a blood transfusion, you may put you in danger. So these are things you want to think about as a traveler and as a citizen who has public health awareness.
So I think we're at the end of our scheduled time. Is that correct? I'm looking at our folks, yes? Or can we take one more question? One more question? Okay. Anybody? Yeah.
Hi. So you've generated such an amazing body of work and have obviously done a lot. So what advice would you have to a young scientist who also wants to do this type of work and become as prolific as someone like you?
Oh, that's very nice, yeah. Aren't I on your committee or something? I don't know. No, really, you know what? You have to maintain the attitude. There's two things how I've done my life is I've never made any decision for my career based on money. Scientists don't get paid a lot, is what you want to know. And then second, don't take no for an answer. That includes number 3, don't give up. So, and lean on your colleagues my colleagues have helped take care of me through my career and my highs and lows. The highs, don't get too full of yourself. And the lows, they help pick you up off the floor when your grant gets rejected. So all that together will make a successful scientist.
So please join me in thanking Michael Gale again for a wonderful talk. And congratulations again. Thank you.